There is a second alien Earth hidden in plain sight. The lab alarm cut through the quiet, and the petri dish marked impossible life wouldn't die. Bleach, heat, UV, still metabolizing. Check the labels, someone said. We did. Same result. Then the whisper, none of us wanted to say out loud, shadow biosphere. If our cleanest protocols can't kill it, what else are we missing? Because here's the real fear. If our tests are blind on Earth, what do we overlook on Mars? This isn't sci-fi, it's a crack in our instruments. And it starts with failed experiments, desert microbes that refuse to be cultured, and a lake that almost convinced NASA life could rewrite chemistry. To see what's hiding, we have to ask a blunt question. Are we only detecting life that looks like us? Not humans, our biochemistry. The life every high school chart recites. Carbon-based, water-using, running on the big six. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, same chirality. Left-handed amino acids, right-handed sugars. It's neat, tidy, and blinding. Now flip the premise. What if some life here runs on alternate rules? Mirror-chiral building blocks, different backbone polymers, strange solvents. It would be here, but our methods would act like it isn't. That's the shadow biosphere idea. A coexisting, non-interbreeding realm of weird life that standard assays literally don't see. How would we miss it? Start with our habits. We grow life in flasks and on plates built for what we already know. It's the Great Plate Count Anomaly in action. Under the microscope, you can count cells. On the plate, almost none appear. In many environments, about 99% of observed microbes won't grow on standard media. That's not a footnote, it's a cliff edge. Then we sequenced. PCR is our gatekeeper. But PCR primers are keys cut for one lock. If a genome used mirror-chiral nucleic acids or a different backbone, PNA, TNA, pick your alphabet, those primers wouldn't bind. The machine would say nothing here. Not because nothing's there, but because our key doesn't fit. So here's what we're going to do. We'll rewind to the Mono Lake arsenic saga, the 201 claim that a bacterium swapped phosphorus for arsenic, and see why the headline crashed and what it taught us. We'll go underground, kilometers down, to the deep subsurface biosphere that could hold roughly a fifth of Earth's biomass, where rock eaters breathe geology's slow clock. We'll walk the Atacama, crack salt crusts, and peer into halite that keeps secrets for centuries. We'll build new eyes, label-free spectroscopy, chirality sensors that can flag mirror life, and sequencing approaches that bypass PCR's bias. Along the way, we'll ask why medicine might already be missing pathogens, or allies, and we'll press the stakes. Planetary protection. If Earth hides a second biochemistry, a Mars rover could be staring at alien life and returning a blank test. The big question, are we alone on Earth? How we became blind, the great filter of our tests. To understand our blindness, we need to go back to a simple rule that became a cage. If it grows on a plate, it's real. Koch's postulates built modern microbiology, but they also trained us to equate culture with truth. That worked, until it didn't. Here's the moment that should have haunted us. You take a scoop of soil, you stain it, put it under a microscope, and count cells. Teeming. Then you spread that same sample on a petri plate with textbook nutrients. After incubation, you get a few colonies, sometimes 100-fold fewer, sometimes 1,000-fold fewer. The discrepancy was named the Great Plate Count Anomaly. In many environments, on the order of 99% of microbes visible by microscopy never form colonies on standard lab media. People called the unseen majority microbial dark matter. And then we shrugged and moved on. We shouldn't have, because culture is only the first filter. The second is molecular, PCR. It's our miracle tool and our blindfold. PCR needs primers, short sequences that bind to a known target. That means it presumes you already know the alphabet and the grammar. Switch the alphabet. Use a different sugar, reverse the chirality, alter the backbone, and the primer doesn't bind. No binding, no amplification. The readout looks like a negative. If a shadow biosphere used right-handed amino acids or non-canonical nucleic acid backbones like PNA or TNA, our universal primers would glide past like they were searching the wrong side of a mirror. We'd file the sample as inert or contaminated and throw it out. Even our stains and probes play favorites. We call them universal because they hit conserved targets in known life. But universal to us just means broad within our branch. A different branch, especially one with flipped chirality, won't take the stain, won't light up the probe, and our microscope turns a living micro-ecosystem into a blank field. Then there's sterilization. We design kill protocols around what our biochemistry survives and what it doesn't. Heat, bleach, UV, ethanol. We have documented survivors, organisms that ride out autoclaves inside protective niches, spores that weather slugs of disinfectant, cells that repair shredded DNA after UV barrages. We tell ourselves they're outliers. But selection works both ways. Our sterility routines might be systematically selecting for life forms, fundamentally harder to disable because they don't share our weak points. If a membrane uses alien lipids, if proteins fold with the opposite-handedness, if the genetic polymer shrugs off hydrolysis patterns we rely on, our clean room becomes a training camp for the unfamiliar. There's a case that still bothers people who work with rocks. Desert varnish, those dark manganese-rich skins that paint arid stone, has microbial fingerprints. But prep those samples the standard way and the signal evaporates. Tweak the chemistry, lower the water activity, and traces reappear. It looks less like nothing was there and more like we were washing it away with our approach. All this primed us for a headline-grabbing moment that arrived on a winter stage in 2001. A NASA-funded team announced a bacterium from Mono Lake, G-Phage-1, that they argued could substitute arsenic for phosphorus in its DNA backbone. Phosphorus is one of life's big six. Swap it out, and the implications are huge. Arsenate esters are notoriously unstable in water, so the claim landed like a grenade. If true, it would mean at least one organism rewrote a core rule of biochemistry, hinting that multiple biochemical styles could coexist on Earth. The pushback was immediate and technical. Critics pointed to contamination controls, to the chemistry of arsenate bonds, to alternative explanations like arsenic tolerance rather than true incorporation. Independent labs dug in. Careful reanalysis, purifying DNA with stricter methods, running cleaner isotopic and elemental assays, found that the DNA still depended on phosphorus. Arsenic wasn't sitting in the backbone. The grand claim didn't hold. The paper was later retracted or effectively defanged by follow-ups. The organism moved from arsenic-based life to arsenic-tolerant extremophile. For some, that closed the book. But the lesson wasn't that weird life is impossible. The lesson was that our protocols needed to be sharper. The controversy forced better extractions, tighter controls, and a broader playbook for detecting unfamiliar chemistries. It also revealed an uncomfortable truth. Even when we're wrong, we often learn that our instruments were biased to our expectations. Meanwhile, while we argued about Mono Lake, the ground under our feet kept a quieter secret. Drill into continental crusts, descend into deep gold mines and boreholes, and you meet the deep subsurface biosphere. Microbial communities kilometers down, starved of sunlight, living on hydrogen from water rock reactions, radiolysis products, and trace chemicals leached from stone. This realm is vast. Some estimates suggest the deep biosphere holds on the order of 15 to 20% of Earth's total biomass. That's not fringe, that's a planetary compartment. Cells down there divide on timescales that make human history look impatient. Metabolism's idle at the edge of detectability. If you brought those communities into a lab and offered our rich broths, many would never wake. You'd label the tube lifeless. You'd be wrong. Stack the filters together. Culture bias, PCR blindness, not-so-universal probes, sterilization tuned to our vulnerabilities, and a habit of sampling easy places. And you get a picture of systematic myopia. We see what we set ourselves up to see. We miss what doesn't answer in our language. And every so often, a field anomaly or a controversial claim jars us into admitting the blind spot. Here's the pivot. If our methods are a great filter, then the absence of evidence is not evidence of absence. It's evidence of our keys not fitting the locks we keep trying. So we change the keys. We stop requiring lab growth to prove life. We add chirality-aware detectors that can spot right-handed amino acids in unnatural ratios. We lean on label-free spectroscopy that listens to vibrations and masses without asking a cell to wear our tags. We design sequencing that doesn't rely on PCR primers, opening the door, however slightly, to polymers we haven't named yet. Because the alternative is worse. To keep declaring deserts empty, mines sterile, and alien worlds lifeless, only to learn we were surrounded the whole time. The Mono Lake shock? Arsenic life controversy. 3 days after the lab alarm, we were staring at a clip from 201, a NASA press conference where a young geomicrobiologist promised that a lake in California might have cracked open the rules. Mono Lake is a hostile soup. Hypersaline, alkaline, spiked with arsenic. Arsenic is the evil twin of phosphorus. Chemically similar enough to sneak into pathways toxic because it breaks them. So when Felisa Wolf-Simon's team announced a bacterium labeled GPOG1 that seemed to swap phosphorus for arsenic in its DNA backbone, it hit like thunder. Why did it land so hard? Because phosphorus isn't a side note. It's in ATP, in membranes, and in the very rails of DNA's ladder. If a microbe could replace phosphorus with arsenic, then the big six elements weren't sacred. Suddenly, early Earth scenarios where phosphorus was scarce didn't look like dead ends. And for astrobiology, the message felt electric. If life can rewrite chemistry here, maybe it can write anything out there. The claim came with a story that fit the lake. Mono's waters carry arsenic. Phosphorus is lower by comparison. The team grew GPIs-1 in media with arsenic and minimal phosphate, then reported arsenic signals co-located with DNA extracts. The takeaway was bold. The bacterium wasn't just tolerating arsenic, it was building with it. Headlines did what headlines do, and the words alien life started hitchhiking alongside arsenic. Here's where the science fought back. Arsenic esters don't like water, they fall apart. DNA lives in water. Chemists and microbiologists flagged this immediately. If arsenic really sat where phosphate should be, the backbone should hydrolyze into mush. Others asked about contamination. Were the DNA pellets clean, or were arsenic salts sticking around to fool the instruments? And if Mono Lake itself has more phosphate than the paper implied, why would any organism prefer the unstable option? Then the blogosphere lit up. Rosie Redfield, a microbiologist with a sharp eye for controls, tore into the methods and posted her critique openly. Independent labs took samples of GPOG1 and started over. They scrubbed DNA extractions with harsher purification, ran mass spectrometry with tighter calibrations, and pushed isotopic assays to pick up trace incorporations if they existed. What they found was sobering. The bacterium was certainly arsenic tolerant. It could grow in arsenic-rich conditions, but its DNA still depended on phosphorus. Arsenic wasn't embedded in the backbone. When free arsenic contamination was removed, the dramatic signals faded. Later analyses reinforced the same point. No wholesale substitution in core genetic polymers. The paper didn't just bend, it broke. Retractions and corrections washed through the literature. GPOG1 stepped down from biochemical icon to tough extremophile with clever detox and storage strategies. Some people laughed it off as a hype lesson, but that misses the real pivot. The controversy forced a methodological cleanup. Labs adopted stricter extraction protocols to avoid elemental carryover. They paired elemental measurements with structure-resolving techniques rather than trusting collocation. They treated universal assumptions with more suspicion. The specific claim died. The toolkit got sharper. Meanwhile, Mono Lake still mattered. It's a natural pressure cooker for odd metabolism. Long before GPOG1, researchers documented microbes there that could use arsenite in phototrophic pathways, pulling energy from arsenic redox cycles, the way others ride sulfur or iron. So even without DNA made of arsenic, the system showed that life can wire energy flow through elements we mostly treat as poison. The lake was never about proving aliens. It was about exposing our confirmation bias. When data hint at unfamiliar chemistry, we tend to either crown a revolution or slam the door. The better path is slower. Tighten controls, widen the search. There's another lesson tucked inside the criticism. Almost every objection leaned on water chemistry, bond stability, and assay contamination. Valid, essential, and squarely within the rules of our biochemistry. But the shadow biosphere hypothesis asks a different question. What if some life avoids our rules entirely? Not arsenic in DNA per se, but alternative scaffolds, mirror-chiral enzymes, polymers that don't respond to our stains or PCR keys. The point isn't to defend a failed claim, it's to notice how fast our tools claim certainty inside their comfort zone. In the aftermath, something else shifted. NASA and astrobiology circles got warier of culture-based claims and more interested in orthogonal detection, multiple independent signals that don't rely on the same assumptions. That means coupling isotopes with chirality checks, pairing untargeted metabolomics with label-free imaging, and asking samples to reveal function before we assign form. It also means designing experiments where phosphorus, sulfate, and standard organics aren't the only doors we try to open. Mono Lake was the false summit that taught us where the trail actually runs. The bacterium didn't rewrite DNA, but it forced everyone to rewrite protocols, and it planted a stubborn question. If we nearly fooled ourselves with arsenic because our extractions carried the element along for the ride, how many times have we thrown away life that didn't ride with our chemistry at all? The lake didn't give us alien life. It gave us a mirror, one that showed how our methods can make strangers invisible. If not arsenic, then what? Plausible, weird chemistries? Ideas are easy to mock until a field sample refuses to behave. So if not arsenic in DNA, what else could a shadow biosphere actually use? Start with chirality, because it's the quietest way to go missing. All known life builds with left-handed amino acids and right-handed sugars. Flip that. Right-handed amino acids, left-handed sugars, and suddenly every enzyme we own is looking the wrong way. Our proteases don't cut, our antibodies don't bind, our universal PCR primers don't latch, because they expect a right-handed sugar in DNA and RNA backbones, not a mirror. The machine would call it negative. Meanwhile, mirror life could be living off the same carbon we do, just unreachable to our biochemistry. Now step off the DNA ladder. Biologists love ribose phosphate backbones, but prebiotic chemistry has shown alternatives that don't need ribose's finicky stability. PNA, peptide nucleic acid, replaces the sugar phosphate with a peptide-like spine. TNA uses threose instead of ribose. They can store information, base pair, and even outcompete RNA under certain conditions in the lab. If a lineage found a way to keep PNA or TNA as its genetic polymer, protected in brines, bound to minerals, or tucked in ice eutectics, it wouldn't show up on PCR at all. Our enzymes don't know how to copy it, our extraction kits might throw it out as junk. We'd call the sediment sterile and miss a genome humming in an unfamiliar register. Silicon keeps showing up as sci-fi, but the chemistry has slivers of plausibility in the right corners. Silicon-carbon bonds can be stable, organosilicon chains exist at high temperatures, under pressure, or in extremely dry settings. Silicon-based side chains or hybrid organosilicon cofactors might scaffold metabolism. Not silicon life in a cartoon sense, but catalytic frameworks that lean on silicon when water would otherwise sabotage the reaction. If your enzyme toolkit evolved in hot, mineral-rich pores, silicon could be the brace that holds a pathway together, one we dissolve during routine aqueous prep. Then there's sulfur. The iron-sulfur world hypothesis isn't fringe anymore. Ancient enzymes are packed with iron-sulfur clusters. Hydrothermal vents build gradients that look like battery stacks. A shadow community could center its economy on metal-sulfur catalysis, with thiol-rich polymers acting as both scaffold and redox buffer. Instead of ATP ferrying phosphate groups, energy could slide along sulfur switches and iron knots, with membranes built from isoprenoid variants or entirely unfamiliar lipids that shrug off our detergents. We try to lyse those cells with standard buffers and just watch them float. Solvents matter more than we admit. Water is our default, but life can carve out microsolvents inside water's domain. Supersaline brines drop water activity so low that normal proteins seize up, some polymers stabilizing that dryness. Formamide can support nucleic-like chemistry and remains liquid where water freezes. Eutectic ice phases concentrate reactants in tiny veins. If an organism built its core polymer inside halite crystals, relying on a brine microphase or a formamide pocket, our first rinse could erase the entire architecture. Later, when nothing grows, we'd blame the sample, not our solvent choice. Energy capture is the other hinge. Sunscreen biology is everywhere at the surface, but deep down, radiolysis splits water into hydrogen and oxidants. Some bacteria already live on that trickle. Rock eaters breathing geologic time. Serpentinization, where water meets ultramafic rock, floods systems with hydrogen and alkalinity. A shadow lineage could exploit peroxide or superoxide as terminal oxidants, or use perchlorate the way some desert microbes already do. If your respiratory chain is tuned to molecules standard life treats as stress, you're invisible to our respirometers and thrive in places we label too oxidizing or too weird. Here's where it tightens. Every one of these chemistries clashes with a lab habit. Mirror-chiral polymers dodge our enzymes. Alternative backbones slip past PCR and silica columns. Organosilicon and metal sulfur frameworks break under aqueous extractions or standard detergents. Exotic lipids refuse our stains. Brine-locked biochemistry collapses the moment we dilute the sample to something physiological. None of that proves a second tree exists. It does mean the escape routes are real. So the plausible map looks like this. Chirality flips that make our keys useless. Genetic scaffolds that never meet a primer. Catalytic cores that prefer metal and sulfur over phosphate shuttles. Solvent microenvironments that punish water-heavy prep. And energy metabolisms that sip radiolysis and rock-borne hydrogen while our instruments listen for sugar highs. If a shadow biosphere is here, it doesn't have to shout. It just has to. Speak of chemistry, we've trained ourselves not to biochemical bunkers. If weird life hides, it chooses places our tools dislike. Start with hypersaline lakes. Water so salty it's barely water. In these brines, normal proteins seize, membranes wrinkle, and DNA repair limps. Yet communities persist. Halophils stack their interiors with compatible solutes and armor their proteins. Now push further. Inside halite crystals, you find thin, trapped films of brine, microaquariums sealed for centuries. Crack one open, and cells revive after impossible dormancy. If a lineage depended on that microphase, chemistry stabilized by salt squeeze, our first rinse would wreck it. The petri dish would show failure. The crystal was the culture all along. Follow the salt underground. Deep salt mines carve through deposits that lock water activity to levels that frighten enzymes. Here, chaotropic stress bends the rules. Standard lipids leak, proteins misfold. But some organisms adapt with exotic membranes and stabilizers we barely recognize. If a shadow lineage built its information polymer to thrive, where water's grip is weak, our aqueous extractions would dissolve the evidence. Label-free spectroscopy might whisper metabolism, but our stains would shrug. Now drop kilometers into the crust. In deep boreholes and gold mines, the deep subsurface biosphere breathes at a geological pace. Hydrogen from serpentinization and radiolysis drips energy into fractures. Carbon trickles from carbonate veins. Methane bleeds from rock. Cells divide so slowly that growth curves look flat lines. Some estimates say this realm holds around 15 to 20% of Earth's biomass, a subterranean reservoir we barely sample. If life deviated early, this is where relic lineages could have persisted, isolated by rock, tuned to chemistry stable over eons. Try to culture them on rich media, and you shock them. Try to amplify with PCR, and you assume a backbone they may not own. Down here, negative result usually means you asked the wrong question. Swing to hydrothermal vents and serpentinization zones. At alkaline vents, natural chimneys build proton gradients across thin mineral walls. Electrochemical batteries that look suspiciously like the first metabolism's training wheels. Iron-sulfur clusters assemble on catalytic surfaces. CO2 is reduced without sunlight. In serpentinizing systems, pH soars, hydrogen gushes, and organics can form abiotically. If any environment incubates non-standard scaffolds, thiol-rich polymers, metal-sulfur circuits, unconventional lipids, it's a pore in these chimneys. Yet we sample vents with water-heavy kits, detergent lysis, and primer sets that demand ribose phosphate. We may be rinsing away the very scaffolds we seek. Step on to the Atacama Desert, one of Earth's driest places and a Mars mirror. Salt crusts there hide microhabitats where perchlorate and nitrate rewrite redox rules. You can detect DNA sometimes, but growth remains faint and unknown compounds dominate chemical scans. If metabolism relies on oxidants, we classify as stress. Our spirometers read silence. Our culturing bottles add water activity that breaks the native solvent balance. The community was alive before we helped. Head to permafrost and subglacial brines. Cold doesn't just slow chemistry, it reorders it. In eutectic ice, salts concentrate into liquid threads, where diffusion shrinks and reactions meet. Formamide stays liquid at low temperatures and can support prebiotic steps. If polymers that look fragile at room temperature become stable in these veins, a cold biosphere could run on a set of rules we erase by thawing. We melt cores, warm filters, and watch signatures vanish. Turn to desert varnish and manganese-rich crusts. These thin coatings act like catalytic skins, hosting microreactors where trace oxidants cycle and metals shuttle electrons. Standard prep, acid washes, oxidative cleanups, can strip the very compounds that signal life, leaving a surface that reads geological. When researchers dialed down water and changed extraction chemistry, faint biological fingerprints reappeared. That looks like a methodological ghost story. The creature was only invisible because the light we used scared it away. Don't skip the basaltic ocean crust. Beneath the seafloor, fractures host biofilms that operate on energy budgets so thin, our models say can't sustain growth. Yet the films persist. If these communities run ultra-slow cycles or rely on mineral-mediated catalysis we don't measure, then our short incubations and sugar-rich assays misjudge them. We ask for rapid reproduction. They offer maintenance and repair. So where do we look? Places that punish water's dominance. Hypersaline brines, salt crystals, alkaline vents, permafrost veins. Places where energy arrives as gradients and radicals, not sunlight and sugar. Serpentinization zones, radiolytic fractures, manganese skins. Places are prep ruins, sites where dilution, detergent, heat, or pH shifts erase structure. And we look with new eyes first. Label-free spectra before lysis. Chirality checks before sequencing. Solvent-preserving microfluidics instead of rinse and repeat protocols. If a shadow biosphere exists, it's not hiding from curiosity. It's hiding from our habits. New eyes, building tools to detect the undetectable. If our habits make life vanish, then the first rule is simple. Look without asking biology to meet us halfway. Untargeted metabolomics is where that begins. Instead of searching for known molecules, we run high-resolution mass spectrometry and let the sample speak. Peaks rise for compounds we can't name. Odd mass defects, strange isotopic envelopes, patterns that don't map to the usual databases. In hypersaline brines, these unknowns cluster around windows where our standards go silent. That's a hint, not a verdict. But it's the kind of hint you only get when you stop forcing the answer key. Next, we refuse labels. Probe-free microscopy, Raman and infrared, reads vibrations of bonds directly, no stains, no antibodies. Raman can pick up sulfur stretches, metal ligand modes, even subtle shifts from different chiral environments. In a vent chimney thin section, you scan across a pore and watch signatures swing from carbonate to thiol-rich complexes to lipids that don't match any catalog. The image isn't a pretty fluorescence overlay. It's a chemical topography, unfiltered by universal dyes that never were universal. Pair that with mass spec imaging, MALDI or nanosimps, and you start layering maps of elements and molecules without ever cracking a cell. Chirality is the quiet giveaway. Life favors one hand. Mirror life would tilt the other way, so we build chirality detectors into the first pass. Circular dichroism spectroscopy tells you if a soup leans left or right. Proteins, sugars, even aggregated polymers have fingerprints. Then we get quantitative. And enteoselective chromatography splits left from right amino acids and sugars with columns lined by chiral selectors. Run a soil extract, and if the DLR ratios blow past biological norms in a consistent pattern, not random racemization but a living skew, you've got a flag. Not proof of mirror life, but a signal strong enough to change the plan for the next drill core. Sequencing has to lose its gatekeeper. PCR was the lock we made. Now we set it aside. Primer-free approaches start with random hexamer priming across all extractable polymers, then capture whatever templates accept a generic handshake. Nanopore devices help here because they don't need synthesis by DNA polymerase to read a strand. They listen for disruptions in ionic current as molecules traverse the pore. If the polymer isn't DNA or RNA, the read may look messy, but the error isn't useless. It's a signature. You can calibrate with synthetic standards, PNA, TNA, xenonucleic acids, to learn how non-canonical looks in the current trace. Add enzymatic traps. Expose extracts to canonical nucleases and watch what resists digestion. What survives might be noise, or it might be the genome you've been throwing away. We can bait the hook with synthetic biology. Build bioreactors that feed on substrates standard life ignores. Offer arsenite as an electron donor while starving phosphorus. Provide formamide-rich phases. Drip peroxide at controlled levels, or spike media with silicon-bearing organic stable only at high pH. If growth, redox changes, or gas exchange shifts appear under forbidding conditions while control cultures flatline, the result demands attention. You're not proving a second tree. You're isolating metabolisms misaligned with ours. Then you flip the test, add canonical nutrients, and watch the signal die. If metabolism fades when conditions become normal, you've probably been normalizing it out of existence. Isotopes give you a lie detector. Biology prefers lighter isotopes. Fractionation leaves a trace, but we widen the lens. Instead of only carbon and nitrogen, we track sulfur, iron, even arsenic in systems like Mono Lake, while combining isotope patterns with chirality and untargeted metabolomes. 3 orthogonal hints speak one loud maybe. That's the astrobiology shift post-mono. No single sensor gets to declare life. You want concordance across tools that don't share the same bias. Field protocols must stop scrubbing samples clean. Microfluidics can preserve native solvents, keep brine briny, keep pH extreme, keep pressure high. We build chips that sip from a fracture and route fluid through Raman, CD, and enantiochromatography without dilution. Optical tweezers corral cells or particles into analysis lanes without detergents. If you must lyse, you do it in situ inside a sealed channel while capturing headspace gases for analysis. The sample never sees physiological unless it was born there. We also need negative space controls, blank rocks, sterilized brines, mirrored chemistries to catch our own artifacts. And we publish raw spectra, not just processed peaks, so other teams can push different algorithms against the same data. A false positive like GPIGE1 gets stripped fast in that light. A real anomaly grows stronger as independent signals pile up. Finally, we practice on Earth for space. Build life detection assays that survive Mars levels of perchlorate and salinity. Test Europa analog brines under pressure and cold. Insert chirality checks into rover payload mockups. Prioritize primer-free, label-free detectors that don't assume ribose phosphate. Bring the deep biosphere's patience into mission timelines. Slow incubations, long watches for faint gas shifts, acceptance that metabolism might pulse on a schedule our shifts don't catch. New eyes aren't one instrument, they're a mindset. Never let a single assumption stand between you and a living chemistry you haven't met yet. Micro stories from the edge, field discoveries that don't fit. While that was happening in clean rooms and conference halls, the strangest clues turned up in places that don't care about our theories. Get this. One clue was found a mile underground by miners who weren't looking for anything living at all. In a South African gold mine, a fracture seeped water older than our civilizations. Down there, no sunlight, barely any oxygen, just rock and radiation. Yet a bacterium was thriving on the products of radiolysis. The split-second chemistry when natural radiation cracks water and frees hydrogen. It wasn't fast. Divide once in years, maybe decades, but it lived. From their perspective, metabolism was a whisper. To us, it was a message. If you expect sugar rushes, you'll call this silence. Meanwhile, the deep biosphere, potentially around a fifth of Earth's biomass, goes on breathing geology. 3 days earlier, something strange occurred in the salt mine lab. A grad student cracked a piece of halite, and an air bubble the size of a pinhead gleamed inside. They wetted the shard with brine and waited. Nothing for an hour. Then, slow green. Cells roused from a glass coffin, reviving after centuries sealed in salt. They didn't rush into our media, they recoiled from it. Shift the salinity even slightly, and the signal died. The pattern wasn't dormancy waking to our kindness, it was dormancy disturbed by our chemistry. We've been taking these crystals home, rinsing them clean, and reporting no life. The life had been inside, contingent on a solvent we always destroyed first. Unknown to them, across the Pacific, a team in the Atacama Desert was nudging open a salt crust with a sterile blade. The instrument said DNA was present, barely, but growth assays stayed flat, and the chemical scans were cluttered with compounds that didn't match familiar databases. Perchlorate hovered in the background, an oxidant most life treats as stress, not food. The crew switched to label-free spectroscopy and saw weak signatures that rose and fell with dawn and dusk humidity pulses. When they tried to help with water and nutrients, the signals collapsed. Everything was about to change when they realized their well-intentioned dilution erased the native microsolvent that made the metabolism possible. Meanwhile, something else was happening at Mono Lake. GPIGE1 had lost its crown, but other microbes there soaked in arsenic like it was weather. Their tolerance was off the charts, their detox pathways not fully mapped. In one microstory, a researcher sampled a mat that refused to behave in the lab. Elemental scans screamed arsenic. Sequencing was anemic. When they ran enantioselective chromatography, the amino acid ratios drifted from expectations. Not mirror life proof, but a nudge that the community's chemistry leaned in odd directions. The sample stabilized only when the team rebuilt the lake's extreme pH and salinity inside a microfluidic chip. Once again, the petri dish had been the problem. According to drill ship logs, basaltic ocean crust hosted another puzzle. Biofilms clung to fractures where energy budgets were so thin, our models said maintenance was barely possible, let alone growth. Yet isotope signatures hinted at active fractionation, and nanosims maps showed islands of enriched carbon tucked against iron-sulfur minerals. Researchers extended incubations from days to months. The readouts didn't spike, they drifted. Gas exchange ticked like a slow second hand. If your instrument expects a sprint, you miss the marathon happening in the corner. Here's where it gets crazy in a hospital basement. A surgeon's team kept finding biofilms on an explanted implant that laughed at standard sterilization. Cultures came back negative. PCR panels drew blanks. The materials lab swabbed the film and ran ramen. The spectrum was rich. Lipids, some sulfur stretches, and an odd shoulder they couldn't place. When they treated the sample with canonical nucleases, nothing changed. But when they ramped salinity and added a peroxide pulse, a faint oxygen uptake appeared. The team didn't claim alien life. They did change their cleaning protocol, swapping one disinfectant for another and altering surface chemistry. The biofilm stopped coming back. Was that a shadow organism or an ordinary bug with an extraordinary shield? The case won't say. The lesson was louder. Our hospitals might be selecting for chemistries our tests ignore. While that was happening in clinics, a field geochemist in a desert canyon ran into varnish that defied the usual prep. Standard acid washes stripped everything down to geology. A colleague whispered, "Try less water, less heat." They did. Suddenly, faint organic signals and manganese redox gradients lined up across micrometer layers. Not proof of other life, but evidence that our prep had been the eraser. Each of these moments carries the same stakes. What's at risk are the lives we label absent, the pathogens we don't see, the biotechnologies we never borrow, and the planetary samples we misread. Action by action, cracking a salt crystal, extending an incubation, switching from stains to spectra. We watch stories emerge that don't fit our catalogs, and the impact is cumulative. Not a single smoking gun, but a shelf of puzzles that lean in the same direction. Either extreme life is stranger than we admit, or some fraction of it is other life we're not equipped to hear. The shadow within hypothesis, coexisting but non-interbreeding life. Each clue pulled us to the same edge. Maybe we're not just seeing extreme versions of us. Maybe we're seeing neighbors, close enough to share the planet, far enough to slip past our tools. That's the shadow within idea. A coexisting biosphere that doesn't interbreed with known life because its chemistry won't allow it. Start with what that means at the ground level. If your amino acids are right-handed where ours are left, our enzymes can't digest you and yours can't eat us. If your genetic polymer isn't ribose phosphate, our polymerases can't copy it, and your repair enzymes look at our DNA the way we look at nylon. Interesting, but not food. Gene flow doesn't cross that gulf. Horizontal transfer, microbes swapping tricks at will, stops at the border. Two worlds on one rock sharing minerals and gradients but not genes. Ecology would keep them apart even more. Known life races toward rich niches, sugar pulses, fresh organics, oxygen streaks. A shadow lineage would lose those sprints, so it settles where our metabolism stumbles. Hypersaline brines, alkali vents, permafrost veins, radiolytic fractures, manganese skins, micro niches we visit with rinse bottles and hot buffers, then call sterile when nothing of ours blooms. Meanwhile, they'd structure themselves around nutrients we ignore, perchlorate instead of oxygen, peroxide instead of nitrate, formamide pockets instead of bulk water. Communication would be cross-invisible too. If signaling rides chiral ligands and unfamiliar lipids, their chatter would pass right through our eavesdropping. Why didn't one side wipe the other out? Because competition isn't one race, it's many. Think of overlapping cities that run on different currencies. In our city, phosphate is money, ATP is payroll, and ribosomes are factories. In theirs, money might be sulfur switches, payroll rides, thiol networks, and factories bolt together on mineral scaffolds that break when we clean them. We don't even tax each other efficiently because toxins cross poorly between biochemistries. Our antibiotics look like graffiti on their walls. Their oxidants look like weather to us. So how would we know they're here? You don't wait for a handshake, you look for fingerprints. Non-standard chirality ratios are the first flag. Terrestrial amino acids skew left, sugars skew right. If you sample a micro-niche and see a consistent flip, right-leaning amino acids stack beyond what racemization can explain, or left-leaning sugars in fresh formation, you're staring at a living process that prefers the other hand. Lipids are the next flag. Standard phospholipids and archaeoisopronoids follow families of branching and ether-ester linkages we've cataloged. Find stable membranes built from unfamiliar backbones with mass spec signatures that don't sit in any known biosynthetic pathway. And you've got a second hint. Then isotopes. Biology tugs lighter isotopes into biomass, but the exact fractionation depends on enzymes and conditions. If carbon, sulfur, or iron isotopes show coherent patterns that don't match known metabolisms across a gradient, say along a vent chimney wall, you've got a third, orthogonal clue. Here's the catch. You can't hunt them with our media. The test design has to respect the border. Run parallel growth experiments, where you intentionally forbid what our life requires. Pull phosphate to trace levels. Replace oxygen with controlled pulses of peroxide or perchlorate. Maintain native salinity, pH, and pressure. Offer carbon sources that resist easy fermentation. Then wire the reactors with label-free sensors, Raman for bond vibrations, CD for chirality drift, enantiochromatography on, the outflow. If signal rises under forbidden conditions and collapses when you add canonical nutrients, you've trapped a metabolism that doesn't like our city's currency. You haven't proved a new tree, but you've fenced off our bias. Meanwhile, in the field, you treat micro-niches as sovereign. Don't thaw permafrost before measuring. Don't dilute brines before scanning chirality. Keep chips sealed, route fluids through optics without adding stains. When you must break cells, try enzymes they've never met. Broad chemical lysis, mechanical shear, supercritical CO2. Then test digestion resistance against our nucleases and proteases. If polymers survive canonical cutters but fall to non-biological cleavages, you've got more than dirt. The last piece is gene flow, or its absence. In mixed communities, known life trades genes fast. If you track metagenomes around a suspected niche and see horizontal transfer drop to near zero across a sharp chemical boundary, while function persists on both sides, you're watching two economies that don't exchange parts. Add time. If the boundary holds across seasons, floods, and lab perturbations, the wall is probably biochemical, not just geographic. This isn't a hunt for a mascot microbe. It's a framework for seeing a society. And it circles back to stakes. In hospitals, a shadow lineage could build biofilms our disinfectants politely ignore. Not because they're superbugs, but because we're throwing the wrong wrenches. In wastewater and soils, they might chew pollutants. Standard microbes can't touch or generate byproducts we misread as abiotic. In space, they're the nightmare and the dream. The thing a Mars rover could miss, and the reason planetary protection has to shift from killing everything to detecting what our kills don't reach. If the shadow within exists, it didn't arrive with fanfare. It survived by being the kind of neighbor our senses are trained to overlook. The test now isn't whether we can imagine it, it's whether we can build a science that stops asking every stranger to carry our passport. Hidden pathogens, or hidden allies. If the shadow within lives beside us, our hospitals and clinics are its unintended playground. Diagnostics are built like locks. They only open for keys we've already cut. Start with how we identify infections. Most panels lean on PCR. Primers tuned to ribose phosphate backbones and familiar genes. They're brilliant at finding cousins. They're helpless if the target doesn't share the alphabet. A pathogen with mirror-chiral nucleic acids or an alternative backbone wouldn't bind the primers. The report prints no organism detected, while the patient's fever climbs and the biofilm spreads. Culture isn't better. Clinical labs plate samples on a handful of rich media under standard salinity, neutral pH, and oxygen levels we call physiological. That's a theme park for our biochemistry, not a stress test. If a contaminant thrives in hypersaline microfilms or breathes peroxide instead of oxygen, those plates are deserts. The petri dish still decides what exists, So we call the weird stuff sterile inflammation, idiopathic, or device failure, and move on. Meanwhile, the film on a catheter or implant laughs at our playbook. Sterilization adds a twist. Hospital disinfection is optimized to cripple our membranes, denature our proteins, and shred our DNA. But selection doesn't stop at the OR doors. If a lineage relies on lipids unlike ours or folds proteins with opposite chirality, the same bleach that wrecks our enzymes might slide off theirs. UV designed to target canonical nucleic acids may underperform against polymers that don't photolyze the same way. Each cycle quietly filters survivors that don't share our vulnerabilities. We read the absence of culturable cells as success. We rarely run label-free spectra on the residue we can't explain. Here's where biosurveillance fills the gap. Metagenomics in hospitals revolutionized outbreak tracing, but it still assumes canonical genomes. We spot MRSA's fingerprints because its pages are written in our script. If an opportunist writes in a different hand, shotgun sequencing tosses its strands as junk. Analysts filter low-complexity reads. Pipelines clean contaminants. The outliers vanish before anyone asks if they're real. That's comfortable. It's also how a shadow microbe could ride an ICU circuit unnoticed. Now the stakes. Imagine a cardiac implant that grows a film. No stain labels, no primer amplifies, no culture grows, yet slowly drives inflammation. Surgeons replace the device. Pathology calls it sterile. The patient returns months later with the same problem. In another wing, an oncology ward sees odd drug responses that don't line up with known microbiome profiles. The panels come back bland, the side effects don't. We chalk it up to genetics and chance. But what if there's a missing partner? Organisms our assays don't see quietly modulating immunity and drug metabolism. Flip the fear to opportunity. Hidden allies could be everywhere we wrote off as inert. Enzymes that shrug at peroxide could detox wounds where oxygen therapies fail. Polymers stable in salt or oxidants could coat implants better than anything we make now. Antibiotics from lineages that don't trade genes with ours might dodge resistance entirely. If you want heat-proof catalysts, solvent-proof coatings, or antimicrobials that don't collapse in bleach, you go shopping in chemistries we've been scrubbing away. So what would medicine need to change? First, parallel diagnostics that don't force a passport. When a culture and PCR panel read no organism, run an orthogonal lane, label-free Raman or IR on fresh, unwashed samples. Chirality checks on amino acids in the biofilm matrix, enantioselective chromatography to spot right-leaning skews beyond racemization. If you see coherent chemical life signs without genetic hits, don't throw them out. Route to specialized analysis that uses primer-free sequencing and enzymatic resistance profiling against canonical nucleases and proteases. Second, rethink sterility as spectrum, not switch. Pair disinfectants with different mechanisms. Oxidants that target thiols, agents that fracture unusual membranes, validated against chemical fingerprints, not culture drop. Run challenge tests in native-like solvents. High salt, high pH, or oxidant-rich microenvironments, not just buffered water. If a residue resists two orthogonal chemistries and still shows metabolic gas shifts, oxygen uptake, hydrogen release under controlled pulses, you have a real adversary, not a ghost. Third, update microbiome pipelines. Keep raw reads. Lower the filters that discard junk. Add nanopore lanes for polymer agnostic scanning. Flag consistent, unlabeled signals across patients with similar syndromes, unknown metabolites, odd isotope skews, or DL-L ratios that defy diet and decay. Treat unassigned but recurring as leads, not noise. Finally, build clinical moonshots where failure counts. ICU cohorts with stubborn device. Biofilms get split testing. Standard of care plus a shadow lane protocol that preserves native chemistry. Publish the negatives. If nothing shows, we've fortified the walls. If signals emerge, slow respirometry under peroxide. Chirality tilts that hold across rooms. You found medicine's blind spot and its next frontier. Hidden pathogens or hidden allies. Either way, the cost of not looking is the same. We keep fighting the wrong enemy and missing the friend in the corner. Planetary protection. Mars, Europa, and false negatives. While hospitals wrestle with unseen neighbors, space agencies face a harsher version of the same problem. You only find what your tools are built to notice. Mars taught this the hard way. In 1976, Viking landed with life detection kits that assumed Earth's metabolism. Add water, deliver nutrients, watch for labeled gas. One experiment, the labeled release, lit up. It saw gas production after nutrient addition and a stronger signal after a second dose. Another experiment looked for organics and reported almost none. The verdict became ambiguous, then softened into probably chemistry. But look at it through the shadow lens. If the soil hosted oxidants that double as electron acceptors, or metabolisms allergic to added water, the first test could have tickled life, while the second erased the evidence. We wrote the contradiction off as rocks breathing. The possibility that life breathed differently never got a fair trial. Fast forward to Perseverance. It's caching samples for return, and the payload is the best we've ever flown. Still, bias sneaks in. We target organics using Earth-centric markers. We sample ancient lake beds because water once pooled there. Sensible, but comfortable. If Mars biology settled into perchlorate-rich niches or brine veins that punish dilution, our handling protocols could flatten the very gradients they need. If its polymers shrug at our standard lysis and refuse our universal primers, clean room labs might process the most important sample in history into a meticulously documented null. Europa and Enceladus sharpen the stakes. Their oceans live under ice, spiked with salts and unknown solvents, pushed by pressure we barely mimic. Plumes jet through freezing vacuum. We fly through and sniff organics, pH, and salts, then infer habitability. But if life there runs on solvent microphases, thiol-rich scaffolds, or lipids evolved for cryobrines, our instruments that assume ribose phosphate backbones and earth lipids will play the wrong scales. Circular dichroism and enantioselective chromatography, tools that can catch a chirality tilt, aren't luxuries on these missions. They're the only cheat code we have against mirror-life blind spots. If plume amino acids lean the wrong way in coherent patterns, that's a living flag you can see from orbit. If they don't, you still haven't ruled life out. You've only ruled out life that writes like us. Then comes contamination, the fear that we'll seed another world with our biochemistry or misread theirs as ours. Sterilization protocols assume our vulnerabilities. Dry heat, peroxide vapor, alcohols, UV, radiation, they hammer canonical membranes and nucleic acids. But if a hitchhiker packs lipids that bounce oxidants or polymers that shrug at UV breaks, a small cohort could ride along. Flip the worry. If mirror life exists on Mars, our tests might not detect it, but our microbes might still poison its chemistry, or vice versa, without either side knowing the other is there. Planetary protection can't just be kill everything anymore. It has to be don't break what you can't detect. So policy needs a pivot. Shift from sterilization-only checklists to diversity-aware detection frameworks. That starts with Earth validation. Run mission-grade detectors in analog sites, Atacama salt crusts, Antarctic brines, deep subsurface fractures, without pre-cleaning away native solvents. Bake chirality checks into first contact. CD spectra and DLL amino acid ratios on unprocessed extracts. Add primer-free sequencing attempts, not to read a Martian genome, but to see if anything thread-like transits a nanopore with reproducible noise different from salt. Pair all that with orthogonal life-agnostic metrics, redox disequilibria that reset with diurnal cycles, isotopic fractionations across microgradients, and slow gas exchanges under controlled pulses of oxidants and reductants. No single instrument gets to declare life. Concordance does. Sample return is the moonshot and the minefield. Back on Earth, containment labs are built to trap our pathogens. But if the sample hosts alternative chemistry, the danger isn't infection, it's misinterpretation or destruction. Quarantine must preserve native states, microfluidics that maintain brines, pressure, and temperature, solvent-respecting workflows that route aliquots to spectra before any rinse. The first measurement should be label-free, non-destructive, and chirality-aware. Only then do you escalate, and you never trust a single negative. You require multiple negatives across independent assumptions before you say no life. There's a deeper reason to change course. False negatives don't just delay discovery. They rewrite history in the wrong direction. If Vikings' mixed signals were life whispering under oxidants, generations learned the wrong lesson about Mars. If a Europa flyby misses a mirror skew because nobody flew a chiral sensor, we'll write another chapter of absence across oceans that weren't empty. Meanwhile, the real biosafety risk is asymmetry. We could seed a world with our robust contaminants while failing to notice theirs, or scoop a chemistry home that isn't alive to us but still reacts unpredictably with our ecosystems. Designing for unknown unknowns lowers both risks. Here's the practical checklist teams can carry. Always measure before you modify the sample. Always run at least one chirality assay. Always include a primer-free, polymer-agnostic lane. Always keep a solvent-preserving path intact from field to lab. And always define success as reducing the space of plausible false negatives, not just hitting detection thresholds for familiar life. Because the worst outcome isn't a dirty rover. It's a clean report that taught us nothing about neighbors we didn't know how to see. Origins revisited. One spark or many. To understand what a shadow biosphere would really mean, we have to go back to the start and ask a question most textbooks dodge. Did life begin once, or did it flare up multiple times with different chemistries, with only one lineage winning the surface? Monogenesis is tidy. A single, last universal common ancestor, LUCA, with ribosomes, a genetic code, and a phosphate economy. But universal can just mean the only survivor. LUCA leaves fingerprints all over modern cells. Ribosomes are nearly sacred. The genetic code's assignments are stubbornly shared. Core metabolic pathways lean on iron-sulfur clusters that look ancient. That points to a common trunk. It doesn't prove there weren't other saplings. Early Earth wasn't gentle. Hydrothermal chimneys pumped gradients through mineral pores. Impact winters and UV barrages rewired chemistry on a schedule that would break most startups. If life began more than once, extinctions could have been common, with one lineage finding resilience in ribose phosphate scaffolds and another fading into rocks, or ducking into niches that never faced our sunlit arms race. Think of prebiotic scaffolds as a relay, not a finish line. In lab studies, PNA and TNA self-assemble in base pair under conditions that challenge RNA. They're plausible stepping stones, backbones, that carried information before ribose stabilized as the industry standard. Polyesters and thiol-rich polymers form spontaneously on mineral surfaces, catalyzing redox loops. In eutectic ice veins and formamide pools, different winners emerge than in warm ponds. If early Earth hosted parallel chemical ecologies, it's not crazy that one or more persisted as small, stubborn lineages when RNA, DNA, protein life exploded. The deep subsurface fits that story too well. Kilometers down, conditions change at a crawl. Radiolysis drips hydrogen. Serpentinization breeds alkalinity. Mineral pores offer scaffolds and shields. That's an incubator where an alternative polymer, more stable in low water activity or high pH, could outlast chaos upstairs. A lineage anchored to thiol networks or mineral templating wouldn't compete well in surface sprints, but it could hold a fortress under rock for eons. When we drill, we sample time capsules. We've already seen metabolisms there that operate so slowly, we mistook them for silence. So was there one spark? The data say at least one survived. But one survivor isn't the same as one origin. The genetic code's near universality is often used as proof of a single beginning. Here's another read. Early life shared genes horizontally like wildfire, and a dominant codex steamrolled competitors. Standard life may have assimilated or erased cousins that played by similar rules. Only lineages with truly incompatible frameworks, mirror-chirality, alien backbones, would resist that gene market. Those would be locked out of the swap meet by design, surviving only where our economy couldn't crush them. What would surviving relics look like? Sparse, slow, and specialized. They cluster in microenvironments that stabilize their scaffolds, salt-locked brines, alkaline chimneys, permafrost veins. They'd resist our enzymes and stains, not by stealth, but by not presenting the targets our tools evolved to hit. Their lipids would dodge our detergents, their polymers would shrug at our nucleuses, their amino acids might tilt right when ours tilt left. In isotopes, they might draw different lines through the same elements, leaving fractionations that don't map to our catalogs. If such relics exist, they change more than a tree diagram. They change inevitability, a single origin, hints at a rare roll of the dice. Multiple workable chemistries on one planet push the odds toward abundance. They also change how we read Mars and icy moons. If life can take more than one path here, then alien worlds don't have to mirror our steps to count. A chirality flip in a Europa plume or a non-canonical polymer in a Martian brine wouldn't be a curiosity. It would be the echo of a truth we ignored at home. There's humility in this. Luca's success made us fluent in one language, proud enough to call it universal. But universality is a map, not the territory. If the shadow within is an ancient relic, we're not the only story that worked. We're the story that dominated the surface and the textbooks. The other story might be smaller, colder, slower. It might never build forests or fish. It still matters. It tells us that chemistry has more than one way to cross the gap from reaction to reproduction. And here's the payoff. Origins research gets a new compass. Instead of asking which prebiotic pathway won, we ask which one survived, where, and why. We stop forcing early Earth into a single lane. We design origin of life experiments that protect multiple solvents, preserve extreme pH, and test chirality not as an outcome, but as a variable. We let vents, brines, and ice each host their own relays. If more than one runner crossed the line, we should be able to find their footprints. We've spent a century perfecting one set of glasses. It's time to bring a second pair. The Shadow Biosphere Moonshot, a 10-year plan. If we're serious, we stop dabbling. We build a decade-long program that treats the shadow within like a mission to another world. Because it is. Think of it as sample return from Earth. Preserve native states, run orthogonal detectors first, and publish every null with the same pride as a hit. Year 1 to 2, lock the grid. We assemble a global sampling network that maps Earth's biochemical extremes with intent, not convenience. Hypersaline basins from Mono Lake to the Dead Sea. Deep subsurface fractures in South African mines and Scandinavian boreholes. Serpentinization zones in Oman and Lost City chimneys in the Atlantic. Atacama salt crusts, Antarctic brines, Siberian permafrost, basaltic ocean crust. Each site gets twin protocols, Standard Lane and Shadow Lane. Standard Lane runs legacy microbiology for continuity. Shadow Lane preserves native solvents, pH, pressure, and redox. No rinses, no physiological dilution. Microfluidic chips route fluids directly into label-free Raman and IR. Circular dichroism, enantioselective chromatography, and isotopic scans before anything is cracked open. Year 2 to 4, build the Atlas. We launch an open metabolome and lipidome project for unknowns. High-resolution mass spectrometry flags peaks with odd mass defects and unknown fragmentation. MALDI and nanosims layer spatial maps of elements and molecules. We standardize metadata, exact solvent chemistries, temperatures, pressures, brine compositions. Every spectrum, raw and processed, goes to a public repository with version analysis pipelines. A red team's job is to break claims, rerun baselines, and prove artifacts. We reward them when they succeed. In parallel, we run a global chirality survey. Soil, brine, fracture fluid, varnish scrapings, DLL, amino acid ratios, and sugar-handedness measured in situ or inside sealed chips. The criterion isn't a single flipped sample. It's a pattern that holds across microgradients and repeats across seasons. An anomaly flag system goes live. Green for within biological norms, yellow for skewed but consistent with racemization or deagenesis, red for coherent flips that track living gradients. Red flags trigger targeted expeditions within weeks, not years. Year 3 to 5, rewrite cultivation. We design parallel culturing with forbidden media that deny what our life needs and feed what a neighbor might. Phosphate stripped to trace. Perchlorate or peroxide pulsed as terminal oxidants. Formamide, rich or high salinity microphases, stabilized inside halite, like matrices. Alkaline, high pH reactors templated with iron-sulfur minerals. We don't chase turbidity. We wire reactors with label-free sensors, gas exchange, and slow respirometry calibrated for geologic tempos. Growth is redefined as function that persists and resets. A Raman thiol band strengthening over months, a chirality tilt drifting with controlled nutrient pulses, a faint oxygen uptake that reappears after peroxide cycles. Control lanes add canonical nutrients to test whether the signal collapses in normal conditions. Year 4 to 6, polymer-agnostic genomics. We prototype primer-free sequencing suites, nanopores tuned for broad polymers, with calibration libraries of DNA, RNA, PNA, TNA, and synthetics. We add enzymatic resistance profiling, canonical nucleases, proteases, and lipases, challenge extracts inside sealed channels, while Raman and mass spec watch what survives. We build machine learning classifiers trained on current trace noise to spot reproducible, non-DNA transits. Any polymer that resists canonical cutters but yields structured signals under non-biological cleavage earns an isolation campaign. Year 5 to 7, cross-validation sprints. For every red flag site, independent teams replicate with sealed sample swaps and blinded chemistries. No single lab gets to own a claim. 3 orthogonal signals. Chirality skew beyond racemization, life-like isotopic fractionation across a gradient, and persistent label-free metabolic signatures under forbidden conditions are required before anyone says the words candidate shadow lineage. At the same time, we task a debunk squad to generate abiotic mimics. Mineral catalysis, radiolysis artifacts, contamination. If an effect survives their best counterfeits, it graduates. Year 6 to 8, medicine and industry pilots. Hospitals enroll a shadow protocol for stubborn device biofilms and culture-negative infections. Immediate label-free spectra, chirality checks, polymer-agnostic scans on fresh samples without rinses. Wastewater plants and remediation sites run trials with forbidden media bioreactors baited with perchlorate, peroxide, or organosilicon surrogates to screen for novel detox pathways. Any functional hit, reproducible signal shifts under non-canonical conditions, feeds a synthesis team that rebuilds the chemistry as tools. Oxidant-proof coatings, salt-hardy enzymes, antimicrobials outside our resistance networks. Year 7 to 9, space synergy. We take the most. Diagnostic, Earth-validated assays and ruggedize them for Mars and ocean world payloads. Miniature chirality detectors, solvent-preserving microfluidics, label-free optics, polymer-agnostic nanopores. We stress test in Atacama, Antarctica, and deep salt mines. Planetary protection protocols shift from sterilization only to detect and don't disrupt. Instrumented clean rooms that run chirality and label-free screens on hardware surfaces, and solvent-aware handling for return samples that prioritizes non-destructive first looks. Year 1 to 10, incentives and culture. All data are public by default. We post bounties for new biochemical motifs, cash for independent replication of a chirality flip, prizes for isolating stable, non-canonical lipids, recognition for proving a spectacular claim wrong. We fund negative results and pre-register criteria for candidate versus confirmed, so the field can't drift into wishful thinking. Annual drills rehearse how to communicate a credible detection without hype, and how to retract with grace if controls later fail. Governance matters. An external ethics board tracks bioprospecting and community rights where samples are taken. If we find functional chemistries, we protect niches first, commercialize later. We also publish environmental impact statements, so a hunt for weird life doesn't erase the very gradients it needs. By year 10, success isn't a press conference with a mascot microbe. It's an atlas of extremes with honest baselines, detectors that shrink our blind spots on Earth and in space, and at least a handful of red flag communities that multiple teams can turn on and off with forbidding conditions. If we're lucky, we'll isolate a metabolism that refuses our currency and thrives on its own. If we're not, we'll have built a science that stops mistaking our silence for the world's. Either way, 10 years buys us clarity. About neighbors we missed, or about the real contours of our own branch. Meaning, what counts as us if life comes in 2 flavors? If there's a neighbor running on different chemistry, the first thing that changes is the mirror. Life like us stops meaning life as we define it. It becomes life we share tools with. That's a small semantic shift with a big punch. Our tests, our safety rules, our kinship stories, they're all built around one metabolism. Add a second, and us becomes plural. Start with identity. Humans already live with trillions of microbes that script our immunity and mood. We call them the microbiome and wrap them into self. But that trust is based on familiarity. If a shadow lineage can't trade genes with us and doesn't respond to our molecules, what's the boundary? Is us biology that our enzymes can touch? Or is us any system that metabolizes, evolves, and builds homes in our world? If their amino acids lean right, if their lipids dodge our detergents, Do we exclude them from the living neighborhood because our tools bounce off? Ethics gets tight right away. Discovery turns quickly into extraction. The history here isn't kind. New chemistries become patents, new organisms become factories. But if these lineages only persist in micro-niches we usually destroy, salt veins, chimney pores, varnish skins, then mining them for tricks risks erasing the very conditions that keep them alive. Protection can't wait for charisma. There won't be a panda. There'll be a faint chirality skew in a halite film that took centuries to set. Rights of nature becomes rights of solvent, pH, and rock. Philosophy has to drop the training wheels. Our definitions of life lean on our architecture. Cell membranes, ribose phosphate backbones, ribosomes, ATP. That's description masquerading as essence. A second flavor forces a functional definition. Bounded systems that harvest energy, maintain disequilibria, adapt by heritable variation. If that's alive, then a community that breathes peroxide and brine, that builds with unfamiliar lipids, that hides from our nucleuses but shows growth under forbidding conditions, sits squarely inside the circle. The word alien stops meaning elsewhere. It starts meaning outside our reagent catalog. Culture will lag, then leap. We've told invasion stories for a century, xenomorphs, pathogens, takeover plots. But cohabitation is the plot twist we never wrote. Neighbors we couldn't notice because our senses were biased. The narrative moves from conquest to coexistence. Museums put salt crystals next to dinosaur bones. School kids learn that life's tree has at least 2 trunks on Earth, with one built for bright, wet sprints and another for slow, dry patience. Kinship broadens. If we recognize another lineage here, we stop expecting Mars or Europa to look like us to count. We also stop thinking our biochemistry is destiny. Intelligence, complexity, meaning they might never ride on ribose, or they might. The humility is useful. It breaks the reflex that equates familiar with worthy. Then come the practical ripples. Lab design shifts from universal reagents to solvent-aware workflows as a norm. Medical diagnostics add an orthogonal lane by default when cultures and PCR say no. Planetary missions fly chirality and label-free optics because no detection loses authority when it only covers one metabolism. Engineering gets an upgrade path. Materials that survive oxidants learn from lineages that use oxidants. Catalysts that ignore water learn from brine-locked chemistries. Us becomes a coalition of metabolisms we can learn from, not just a club that shares an alphabet. The last change is responsibility. If there's another biosphere, even a thin, quiet one, we don't get to be careless giants. Our wastewater, drilling, mining, and clean labs can sterilize niches we never meant to touch. So we adopt a precaution built for the unknown. Don't rinse first, measure first. Don't assume your assay is a mirror for reality. Assume it's a key for one door. What counts as us? Maybe it's not a chemical passport. Maybe it's a shared address and a handshake we had to invent new fingers to feel. If life comes in 2 flavors, us is the neighborhood, and the neighborhood just got bigger. If life can hide beside us, then our tests, our missions, and our sense of kinship need a reset. The risk isn't monsters, it's blindness. Want the sequel to this hunt? Mars, false negatives, mirror life detectors, and the hospital case that cracked a lab open? Subscribe, watch the next deep dive on reality's blind spots, and drop your boldest detection ideas below. The best questions shape the next expedition.